Okay, good morning, everybody. And we're going to complete our discussion about the blood today. And then next class, we'll move on to talking about the heart and seeing how the heart functions to pump blood around the body. And then we'll have another class, which will be on the circulation system itself. All these important and relevant topics, especially at the moment with the COVID-19 pandemic. The idea of infection and immunity and everything isn't much on all of our minds. And in fact, COVID-19, one of the things that it apparently does is attack elements of the circulatory system. So I finished off last time with this diagram, summary diagram for you of the different blood cells and just a single basic function for all of them. Some of them have more functions than are delineated here, but these are all that you need to know. The only one I did not mention is this one here, this blood cell called an eosinophil. We're not going to discuss it in much detail because its function is actually quite complex. But one of the things that you can try and remember about the eosinophil is that it's especially designed to deal with parasites, worms, things like this. But the other thing about eosinophils is that they do become involved with the allergic response. I'm not going to discuss how. You just can remember that they, in an allergic response, eosinophils do become involved. Okay, so work through these, make sure you understand and can recognize these, even if you see different diagrams. Okay, you're not necessarily going to see these exact pictures, but you should be able to recognize them by the features of the cell and state one of the functions. Okay, so let me just go back here, and I just want to tell you something. These are what happens to monocytes. Monocytes are in circulation in the blood, but they migrate into the tissues of the body, and they become this important cell, macrophage. And the macrophages are amoeboid cells. They move just like amoebae, creep through all of the tissues, and they are surveillance cells. And when they encounter something which doesn't look right, they alert the immune system. And one of the things that they are particularly responsible for is to govern this set of circumstances which we refer to as inflammation. Inflammation is tremendously important. It is the first real organized response of cells of the immune system to a particular situation, either tissue damage or invasion with a pathogen like a bacterium or something like that. It's a completely normal, healthy response. Even though it may be inconvenient to us, may be painful and inconvenient, nonetheless, it's a sign that the immune system is responding correctly to the presence of something like a pathogen. And it is largely governed by those amoeboid cells, the macrophages. And what they are doing is they are responding to the presence of pathogen, let's say, by releasing hormones. They release substances into the bloodstream which have effects remote from this little tiny cell itself. They can release these hormones in extremely small amounts and yet have an effect over the whole body. So there are 4 signs of inflammation. The first is pain. That pain is due to the fact, well, first of all, there may be a pain response from pain nerves immediately, but in actual fact, during inflammation, the nerves become even more sensitive. They become very, very sensitive, and the pain threshold falls so that what would not normally cause pain at that area now actually begins to really produce a sharp response. And this is due to hormones released by the macrophages. what the idea behind it is that if you have an area of damage, you want to mark that area so that it is not poked at or inadvertently bumped, etc. So the pain there is a marker for your brain. You begin to protect that area, which is advantageous. That's the advantage of having the pain receptors hypersensitive. The next thing that you notice about inflammation is that there is swelling. And the reason for that is, again, hormones released by the leukocytes in the area, especially things like histamine, which cause the blood vessels in the area to become leaky, not to red blood cells, but to the serum. So the serum starts to flood the area, and that causes the swelling, along with the fact that blood vessels actually are beginning to dilate, to get larger. Then the next two are probably the real hallmarks of inflammation that we would notice. And these are that the area goes red and that it gets hot. And that's for two reasons. First of all, the dilation of the blood vessels in the area, that's what you're observing as being the redness. That's one. And then second, the metabolism of cells in that area is actually increased. So they produce more heat. And this actually makes it very advantageous for cells of the white blood cells of the immune system to act. And it disadvantages things like viruses and that sort of thing. They don't like a high temperature. So those are the those are the local reactions of inflammation that produce the typical red, swollen, tender patch that we will observe, say, around the site of an infection by bacteria. But there may also be effects globally for the whole organism. The temperature may be raised, for example. Your temperature may go up. There are a number of different effects on the whole body, which can arise from the small local area of infection, mostly due to the hormones released by the leukocytes. So we have a look at what happens in sequence when, let's say you're working on, you spike yourself on a rose thorn, which has bacteria in it. Well, what have you done? You've caused physical damage to your skin or whatever. And when that happens, those little cells we talked about before, the mast cells that are full of histamine, they are in all of our tissues. They respond to damage or shock by immediately releasing histamine. And histamine is what causes the blood vessels to dilate, and it also causes them to start to become leaky. They begin to leak serum into the area. So masts, that immediate redness, that is due to the action of the mast cells. Macrophages in the area will start to eat both damaged tissue, but also pick up pathogens, and eat bacterial pathogens, for example. And when they do that, they alert the rest, they release the hormones, which alert the rest of the immune system. And the cells, one group of cells which immediately begin responding are the neutrophils. And the neutrophils begin to migrate towards the site of that infection. They're swept around your body by the circulatory system the whole time, but as they approach an area of infection, from which chemical messages are being sent by the macrophages, they will slow down and they will grab onto the sides of the blood vessels. And then they will creep out of the blood vessels and migrate towards the area of infection, where they'll begin to accumulate. At first, they come in, they're very, 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 very vigorous. They rush around and eat up bacteria and as fast as they possibly can. Their metabolism is so high but they actually burn themselves out after a while. They only last a couple of days and then they die. And they begin to accumulate as pus in the area. The macrophages also release hormones which cause the red blood, the red bone marrow rather, to produce more neutrophils, to sway towards producing neutrophils. In fact, if somebody has an infection, an ongoing infection, you can often see if you do a blood slide, you can see that the number of neutrophils in their blood has increased in response to the infection. So neutrophils all move in. Just excuse me, I'm sorry. Try and stop this bonging from my phone. The neutrophils move in. They frantically do all of it, but they accumulate as pus as they die. They don't move out to die. The macrophages, meantime, are releasing all sorts of hormones. They are responsible for causing the nerves to become hypersensitive. They raise the temperature in the area of the wound, but they also may, in the end, actually end up causing the whole body temperature to increase if the infection is ongoing. Once the pathogen is eliminated, which it usually is, this is a very efficient response to this. And they will usually be able to eliminate the pathogen at the site of infection. And once they do, the macrophages do all of the cleaning up. Sometimes an infection can be so vigorous that you may actually accumulate so much pus that it begins to increase the swelling and the pain. And we observe that as an abscess. And in those circumstances where a lot of pus is being accumulated and is causing a pressurized kind of cyst to form, it's useful in those circumstances to actually open up the wound and drain the pus. That we call lancing. So if you have a serious abscess or a boil, lancing it and draining it can be effective. Okay, so let's just have a word about the thrombocytes. The thrombocytes, also known as the blood platelets. And the blood platelets are these very small, they're not really cells on their own. They are living, but they are fragments of cell. They are themselves not long-lived, most probably a matter of days. And their principal function in the body is to promote blood clotting. So I would tell you first of all that in the serum, in the plasma of the blood, a high proportion of the proteins that are present in blood are a protein. It's actually a group. There's several of the proteins, but they together are referred to as fibrinogen. There's the name there. And fibrinogen is in solution in the blood, carries on circulating. It's just part of the serum. When you take, as long as thrombocytes are moving and are not settled, as long as they're in circulation, they are essentially passive. But in response to certain circumstances, for instance, if I slow them down, or if they stop and attach to a surface, then they begin to release this substance called thromboplastin. So that thromboplastin interacts with another substance in the blood, in the serum, also dissolved in the serum, called prothrombin. So thromboplastin, released by the platelets, interacts with prothrombin in solution, and that together acts as to produce this substance called thrombin. So thromboplastin, prothrombin together form thrombin. And thrombin, I know it's complicated, but the thrombin now acts on that fibrinogen, this abundant protein that is present in the serum. And the fibrinogen comes out of solution, and it begins to form long molecules which form a gel. This now, it's semi-solid substance, and that semi-solid fibrin is called, begins to trap blood cells. It traps more platelets, and when the platelets are trapped, they release more thromboplastin, and so it begins a cascade. You start off with just a small amount, and you rapidly build up a large amount of this fibrin with trapped red blood cells, trapped platelets and everything. And that is what we recognize as a blood clot. And just picture, for example, if, for example, you cut yourself, you cut your skin, you sever vessels, usually just small vessels, capillaries that bleed. Now all that blood that is coming out of the capillaries has left the normal circulation, and it will pool. And as soon as it pools, the platelets in it release thromboplastin. The thromboplastin acts together with prothrombin to form this enzyme thrombin, which acts on the fibrinogen. And the fibrinogen now then forms a clot. And that is why, if you look at a cut, it will bleed for a while, but after a while, the blood will begin to form a semi-solid clot, which will actually plug up the cut blood vessels and stop the bleeding. That is the whole idea. You can speed this up if you have a cut. You can speed the process up a bit if you put pressure onto the wound, because what will happen then is that you will cause the blood to stop flowing at the cut capillaries, and a clot will form very rapidly in the cut capillaries and block them. So if you have a cut and you put a pressure on it, you speed up the formation of blood clots in the capillaries and stop the bleeding even quicker. But under normal circumstances, for most people, if the whole system is working properly, the clotting will happen anyway. The clotting will happen naturally. Okay, so, sorry. Let's also have a talk about the blood groups. You've, I'm sure, all heard about blood groups, the fact that different people, members of the population, have various blood groups that they belong to. It's very important. You should know your blood group. You should be able to recite your blood group. Mine is A positive, which is a very common blood group. But there are a number of different blood groups, and they are based on the fact that red blood corpuscles carry on their surface, on their cell membrane, they can carry certain proteins. There are two important ones to discuss first of all, and these are the A protein and the B protein, and they're referred to as A antigen and B antigen. An antigen is anything which will excite an immune response. If you have A antigen on your red blood corpuscles, then your blood group is A. We won't worry about the positive and the negative at the moment. If you have A antigen on your blood corpuscles, it's a protein called the A protein, the A antigen, and your blood group is A. If you have a protein called B antigen, then your blood group is B. But it is also possible for people to have both proteins, A and B, and their blood group then is AB. It's also possible to not have any protein on any of these proteins on the blood corpuscle, and then the blood group is O. So A has A antigen, B has B antigen, AB has A and B, and O has none of them. And those are the four blood groups that are recognized: A, B, AB, and O. Here's the trick. Here's why this is so important. We, in our bodies, produce antibody to the blood protein which we do not have. If you have A in my blood, because my blood group is A, I produce antibody, and I continually have circulating antibody to the B protein. And if you put any of that B protein into circulation in my blood, the antibodies will attack it, and they will cause it all to stick together. It's a very dangerous situation. If I have to have a blood transfusion and they by mistake give me B blood, the anti-B antibody in my blood will attack the B erythrocytes, the B red blood cells, and will cause them to start clumping together, and they'll end up blocking blood vessels, etc., etc., etc. So that's a very dangerous situation. This is why knowing a person's blood group is so important, especially if they're going to get a blood transfusion. So if you are A, you produce antibodies against B. If your blood group is B, you will produce antibodies against A. If you're AB, what happens then? You don't produce any antibody at all. You don't produce antibody. Okay, and if you're O, if your own red blood corpuscles have no antigen on them, have neither A nor B, you'll produce antibody to both A and to B. So this sets up a situation where we can decide if somebody needs a blood transfusion. We can predict what would happen, and we can give them blood which their blood will not react against. So you're always safe, for instance, getting your own blood group. That is one thing. But you may also sometimes be able to use another blood group. So, for example, A blood has antibody to B. So somebody like me with A group, I can receive A blood, obviously, or I can receive blood from somebody with the blood group of O. And I can only donate to A. I can't donate A blood to B. They've got antibodies there. I can't donate to O. I can donate to AB. I should add that in. B blood has antibody to A. It can only receive B blood or O blood. O, it begins to look like it can donate to everybody, doesn't it? Yes, it can. O blood can donate to everybody, and it's called the universal donor. So B blood has antibody to A, can only receive B or O, and can only donate to B. So AB blood has no antibody at all, and can receive. They are called a universal receiver because they can receive blood from any of the blood groups, A, B, AB, or O. O blood can only receive from O because it produces antibody to both A and B. It can only receive from O. So it is a universal donor, but is very restricted in what it can receive, what blood they can receive. Okay, so this little chart explains, I know at first hearing that's very, very confusing, right? But have a look here, and you'll be able to work it out. Here's blood cells with their proteins. Group A has A protein on the outside. What antibody do they produce? They produce anti-B. So the antigen is A antigen. Group B, they produce anti-A antibodies. Group AB do not produce any antibodies at all. And group O produces antibodies to anti-A. It produces against A and against B. It produces anti-A and anti-B. And that explains all of the donation, all of the reasons why people can donate in that fashion. However, there's a complication, and that is that there is another very important blood protein, and that is called the RH factor. But we do not produce antibodies to RH factor unless we have been exposed to a situation where we see the wrong RH. I'll explain. Most of us, most of the population is RH positive. So I am A positive. I've got A group, and I have the RH factor. That is by far the commonest situation. 85% of the population have. But it's the people who we're really interested in are the RH negative population. They are only 15%, and their blood cells lack this protein. They are absolutely fine as long as they are never exposed to RH positive blood. If we are positive, you don't produce any antibody to them, and it doesn't matter. You can receive blood from an RH negative person or from an RH positive person, makes no difference. But if you are RH negative and you are exposed to RH positive blood, the first time you're exposed, you wouldn't notice anything, because it takes a couple of weeks for your immune system to then produce the antibody against the RH factor. But you have that antibody forever. You have a situation where your immune system will remember that forever. And if you again receive an RH positive blood donation, your immune system will react against it very, very strongly and in a very dangerous manner. It will start to cause blood clotting. It's as if the RH negative person, if they receive a RH positive exposure, it's as if they've been vaccinated against RH positive. So if they're exposed again, their immune system reacts extremely strongly. This is now almost never heard of in blood donation, because you are tested when you donate blood or when you're receiving blood. You very carefully establish what is your blood group and what is your RH status. So it's almost unheard of for people to have this situation. But there's one particular situation where this does arise, and that is if a woman is RH negative and she conceives a child which is RH positive, which can happen because they can receive that gene from the father. So the child can be RH, the fetus can be RH positive, while the mother is RH negative. And that can count as an exposure. And the mother begins to produce antibody. And usually the first child that is RH positive is born without any problems. But another child conceived under the same circumstances, RH positive fetus, RH negative mother, the mother's antibodies can move across the placenta and they begin to attack the child's blood. And in utero, inside the uterus before birth, this doesn't cause enormous problems because the fetal circulation allows the fetus to be maintained without, even when there is some damage to its own blood. But when the child is born, it is often born, it's with an ongoing attack by the mother's antibodies circulating in its body against the child's blood. And that is what we call a blue baby syndrome. And it is nowadays, it is dealt with by giving the infant an immediate, complete blood replacement, which drains all of the mother's antibody from the body and replaces it with blood serum, which doesn't attack the infant's blood system. So that is a very dangerous situation when the mother is Rh negative and the child is Rh positive. Okay, so this just gives you the summary of who can donate to who. If you're A positive, you can donate to A positive, obviously, and to AB positive as well. And you can receive from all of these groups, A positive, A negative, O positive, and O negative, and so on and so on. O positive can give to all of these as long as they are positive. And O negative is the one blood group which every blood transfusion service tries to recruit. Extremely rare in terms of the blood groups, but they are able to donate to everybody. These are the absolute universal donor. O negative has no Rh on their blood corpuscles, and they have no protein on their blood corpuscles. So they can donate to everybody. But unfortunately, they can only receive from O negative. If they need a blood transfusion, that's all that they can. AB positive, they can donate to only to AB positive, but they can receive from everyone. So they are a universal acceptor. So just work through this table. It's like a Sudoku. And you work out, when you see this, remember in your brain, what does this blood corpuscle have on it? It has A protein. What is circulating in this person's bloodstream? Anti-B antigen. So that restricts what they can receive. Work out why they can. That is the case. Why is it that they can receive this and they can give this if they have that protein and that antibody? Okay, let's talk about a couple of the disorders of blood. First of all, one of the commonest that you'll hear about is a condition called anemia. And anemia means that the person has low hemoglobin levels in the blood. And that can be due to two things. Either they have enough red blood corpuscles, but there is not enough hemoglobin in the red blood corpuscles, or else that they have very low numbers of red blood corpuscles. Both of those situations will lead to this condition of anemia. There are many, many, many causes for this. And it is more common in women, probably because of menstruation, monthly menstruation. But it can arise in anybody, and it can arise for many different reasons. Severe anemia impedes the ability of the blood to oxygenate tissues, and with obvious effects. The metabolism of those cells is going to be, of all of the body cells, are going to be drastically affected. Characteristic of anemia are things like fatigue, breathlessness, and this sort of thing in severe cases. And there are, in severe cases of anemia, blood transfusion might be resorted to, but often there are other things which can be used. There's supplements, for example, iron supplements, this sort of thing can be used as well to try and balance it. There is, as far as iron is concerned, there is in fact a specific anemia associated with low iron content in the body, and a deficiency like that will result in a low hemoglobin count. But there are iron supplements and also diet. We get much of our iron that we require. We get much of that from green vegetables and green leafy vegetables, and especially from green, slightly bitter vegetables like chard, that sort of thing. There is another kind of anemia called pernicious anemia, And pernicious anemia is due to a vitamin B12 deficiency. This is an essential vitamin, and there are two reasons that you can acquire pernicious anemia, either because you lack vitamin B12 in your diet, in which case it's relatively easy to deal with. You can have vitamin B12 injections and then adjust the diet so that you increase the amount of vitamin B12 that you acquire. But more dangerous is that there are certain examples of pernicious anemia where the person has plenty of vitamin B12, but their body simply can't utilize it. There's a failure to respond, and that is extremely dangerous because it interferes with hemoglobin synthesis in the body. And the pernicious anemia can, in fact, be fatal, especially where it's not a simple deficiency, where the body is simply not able to use vitamin B12. This can arise as a result of something going wrong in the immune system. And it's the result of an autoimmune response. The body's immune system actually attacking the body's own function. The next one is called aplastic anemia, and that is simply because for some reason the red marrow of the bones does not make sufficient red blood corpuscles. There are many, many, many causes for this. But in the one situation where you do see this arising is in people who are being treated, for example, for cancers or something like that, are being treated with chemotherapy with very toxic agents which are intended to kill the cancer cells. And as a byproduct of that, the red bone marrow is attacked and aplastic anemia results. There are certain toxins which we can acquire as well, which will also do this. But probably nowadays, the most common cause of aplastic anemia is chemotherapy. In addition, radiation treatment can also do this. Anything which suppresses the red bone marrow can result in aplastic anemia. And it can be difficult to deal with, but usually is dealt with for the time that people are under chemotherapy, for example. They may be receiving blood transfusions to increase their red blood cell count. Now, the one condition that I do want to spend a little time talking about is this, another form of anemia called sickle cell anemia. Sickle cell anemia is of particular interest in African American populations. It's very, very rarely occurs outside of African American populations, especially here in the United States, because it is sickle cell anemia is a disease which is endemic, particularly to Africa, to West Africa, where certain strains of malaria occur for very peculiar reasons, which I won't discuss. But populations have acquired sickle cell anemia, and it is passed on genetically. So you inherit it from your parents, and you can inherit either from the mother or the father or from both. And the difference in disease presentation if you have only from one parent or if you have from both parents. It's much more severe usually in those who have from both parents. And it's extraordinary disease because it's a response to a single mutation on the gene to produce hemoglobin. So there's a single base change in the DNA sequence, and it changes one single amino acid in the protein hemoglobin. Now, normally, hemoglobin, we've said before, hemoglobin actually picks up oxygen in an oxygen-rich environment, and it will carry it as long as it's in an oxygen-rich environment. When it gets to an oxygen-poor environment, it releases it. It does that by actually changing shape, believe it or not. When it absorbs oxygen, it adopts one shape. When it drops it, it adopts another shape. Just a slight shape change. And if we watch the hemoglobin go back and forth, it just springs back like this. It changes shape continuously, and then it's restored. And to change shape, it's restored. Unfortunately, this amino acid change here means that it doesn't change shape so easily. It will pick up oxygen. Sickle cell hemoglobin will pick up oxygen. It will go to somewhere with a low oxygen. And when it drops that oxygen, it changes shape completely. And it adopts a permanent shape change. And it becomes crystalline. The hemoglobin starts to form like almost like threads together inside the red blood corpuscle. Now, here's how a normal red blood corpuscle functions. The red blood corpuscles circulate through the blood circulatory system, and they move through finer and finer and finer and finer vessels until they get to the periphery of our body, where they're going through blood vessels which are barely the width of a red blood corpuscle. They are plastic. They can deform. The red blood corpuscle can deform quite easily to make its way through these very fine capillaries and then be returned to the heart to be pumped to the lungs to pick up more oxygen. Unfortunately, the sickle cell red blood corpuscles, when the hemoglobin loses its oxygen and it forms these crystalline structures inside the red blood corpuscle, it pulls the red blood corpuscle into a twisted sickle shape like this. And it is fairly rigid and brittle. It can't move through these fine capillaries any longer. And instead, they start to clump on the capillaries. And of course, what happens when they clump like that? They start to slow platelets down. And they start to form clots. They begin to block up these capillaries. Many, many, many of our organs are richly invested with these fine capillaries. Many, many places in our body have forests of these fine capillaries moving through them so that the blood can supply all of our tissues with oxygen. And these capillaries are the ones which start to get blocked. And it can cause agonizingly painful reactions. And it, in fact, can become life-threatening. So sickle cell anemia varies a lot in its presentation. Some people only have it mildly and only rarely have outbreaks of really painful outbreaks. But other people suffer from it constantly, and there are many people for whom it is really, really extremely debilitating. It varies a lot based on genetics, based on when you have one copy or two copy, et cetera, et cetera. Fortunately, I can assure you that virtually everybody listening to this lecture, you are going to live to see a cure for sickle cell anemia. Sickle cell anemia is one of the diseases which we know is very, very amenable to gene therapy. If we can find correct methods to correct this error here, then we can, in fact, see very, very good prospects of curing this disease. So gene therapy is really the real hope of a cure for sickle cell anemia. At the moment, in severe cases of sickle cell anemia, people have actually been cured by having a bone marrow transplant. And that is a replacement of the faulty stem cells which produce this with healthy stem cells, which will then produce normal red blood corpuscles. This is actually relatively common in our African American populations here in the United States, and it's the cause of an enormous amount of suffering. So, but the good news, there is very definitely a cure on the horizon. Okay, so here's a little bit of extra credit for you. Hand in, please, by November 15th, and a 1 to 2 page discussion about what I just mentioned in passing, which is this, the bone marrow transplant. What is a bone marrow transplant? What's the idea of the bone marrow transplant? What is the theory behind it? How does it work? Address those, tell me how a bone marrow transplant is done, and maybe give examples of specific diseases where it has been successfully used. Okay, hand in by November 15th. Hang on, what's happened here? Okay, so let's just have a couple of other disorders and diseases. Things that you may hear about is periodically. First of all, an embolism. An embolism is anything which can cause a blockage in an artery. And the embolisms, most commonly, an embolism is a clot, in which case it's referred to as a thrombosis. But you can also have situations where, for example, you have an air embolism. Air embolisms periodically arise in vessels in the lung, for example. But blood clots are by far the commonest. But you can also have a fat embolism. And this is an extremely dangerous situation. And the one situation where it does occur is when people have a large bone fracture. For example, if they have a long bone, the femur or something which is fractured, and fat from the marrow enters the blood circulation and forms an embolism. This is a very, very, very dangerous situation. The thrombosis, when a blood clot forms in a blood vessel, can result in many different conditions. We'll talk, for example, about thrombosis, a coronary thrombosis. When we talk about the structure of the heart, that is when a thrombosis forms, a clot forms in a blood vessel and blocks the blood vessel of the heart itself. But the thrombosis is also a common cause of stroke. Stroke is a situation where part of the brain is deprived of blood and hence of oxygen. And that can arise from two circumstances. The commonest is from a thrombosis, a blood clot moving into the blood vessels which supply the brain and block them, and then a portion of the brain is starved of oxygen. And that results in severe effects on the body, of course, and that we refer to as stroke. The next condition is one I'm sure most of you have heard of, and that is the condition of hemophilia. Let me just tell you, when I told you about blood clotting, I only told you about it in very simple terms. In actual fact, the substances in solution in the blood, which will result in clotting, there are many, many, many of them, and they're called clotting factors. We don't need to worry about what they are. But each of those clotting factors is produced by a separate gene. And if any of those genes have become mutated and are no longer producing the normal functional product, then the whole system of blood clotting is impacted. The commonest is one factor called factor 7. In fact, factor 7 is the one that most commonly has errors in the gene, and the factor 7 is inoperable. And the entire clotting cascade stops, and the blood will not clot. This is very, very dangerous, because even a simple cut that normally we would clot within minutes and be able to stop, in somebody who is deficient in one of the blood clotting factors, that blood will continue flowing even from that small wound. A bruise, for example, a bruise in most of us, a bruise is where blood vessels get damaged underneath the surface of the skin and bleed underneath the skin. In a normal circumstance, clot forms very quickly there, and then the bruise resolves over a matter of days or weeks. In somebody who has deficient clotting factors, that bleeding continues and continues and continues and continues. And this is a very dangerous situation. And people effectively bleed to death very easily because they simply lack all the clotting factors. These conditions of hemophilia occur predominantly in males for reasons we'll discuss if we talk about genetics later on, human genetics later on. They do occur in females, but far more rarely. They're more common in males, and it's usually treated by giving the person those clotting factors, the missing clotting factor. Also blood transfusion, of course, if they've lost a lot of blood. Lastly, let's talk a little bit about cancers of the blood cells. Remember that all of our blood cells are being produced in the red bone marrow. They're being produced by division of stem cells, which then go on to develop and divide. Whenever we're talking about cells dividing, we open up the possibility that control over that division gets lost for one reason or another. It may be an inherent reason, maybe a cell mutated and its genetics changed, and it loses control over the divisions. Sometimes it's caused by viral infection. Viral infection can cause cells to become cancerous. And cancerous cells, first of all, most are immortal. Second of all, there is very little control over their division. And the progeny of cancer cells seldom develop as they normally should. So there are many of the cells of the immune system, for example, which are subject to cancerous change. And we observe these as particular groups of diseases. The first you've probably heard of most of all, and those are the leukemias. And there are many different kinds of leukemia. But in all of the leukemias, the production of white blood cells, of leukocytes, overtakes the production of red blood corpuscles. And it's observed in a blood slide, for example, you start to see many, many, many immature, non-functional white blood cells appearing in the blood circulation. The leukemias vary tremendously in severity. There are leukemias which are peculiar to children, leukemias which are peculiar to older adults, etc. And some of them are relatively easily treated, and many now are curable. Having leukemia in the old days used to be a death sentence. Nowadays, there are many strategies which can be adopted. And the treatment for leukemias varies a lot, but it usually is a combination of chemotherapy and radiation if need be to irradiate those areas of the red bone marrow, for example, which are overproducing. But it may also respond to bone marrow transplant. So that you can, if you're doing the extra credit, that you can look into. These diseases actually are all related to one another. The next group of diseases are called the myelomas or multiple myelomas. And these are diseases which specifically affect the B cells. B cells are lymphocytes. They are responsible for producing antibody, and they do so by maturing from B cell to the cell called a plasma cell, which actually releases the antibody. And myelomas are cancers of the B cells and of the cells which would produce B cells. Lymphomas are cancers again, of leukocytes, white blood, but those which lodge in the lymph nodes. And so, again, there are several different kinds of lymphoma, but they center on the lymph nodes. And the primary symptom, in actual fact, of lymphomas is that one or more of the lymph nodes begins to swell as it accumulates the cancerous cells, but it's usually painless. Any painless swelling of a lymph node should be examined immediately. They would be biopsied to see if there's any cancerous cells there. Okay, so leave it there for today, and I'll see you then again on Wednesday. And on Wednesday we will start to discuss the heart, and then we'll discuss the circulatory system.